Hello everyone. Good afternoon. I hope you guys are doing good. So let us begin with this session. So we are talking about Cell the Unit of Life. This is a chapter from class 11th. So we'll be doing this chapter. So let us begin. So before I begin this session, let me tell you about our amazing platform that is ODA. So you can learn at ODA and you can click on the icon that is present in the description box. So what you're going to get in the ODA class, you're getting 1 week sessions, three mathematics, three science classes. You also have mentor teacher support. You'll be also getting homework on the application itself. So once the live classes are over, you also get the homework on the app itself. And you also have test series. Yes, and this is just for rupees 29, 49, or 99. It depends upon the class. So what you can do, you can just go on the description and you can join the ODA class now. So before I begin this session, let me introduce myself. Hello everyone, my name is Kamal Pandey. So earlier I was a researcher in the field of cancer biology at NIT Rourkela. So I've been teaching for past few years now. Let us begin with this session. So these are the chapter contents which you are going to learn in this chapter, that is Cell the Unit of Life. So you have this introductory part and the cell theory, prokaryotic cell. So I'm planning to do these three topics in today's class. Okay, so we are going to do the introduction, cell theory, and the prokaryotic cell in today's class, and then we'll be continuing this session on this. We'll be having the part 2 or part 3 then. So you also have eukaryotic cell, where we'll be studying about animal cell and the plant cells. We'll also have introduction about cell membrane, which is also called as the plasma membrane or the plasma lemma. You also have cell wall endomembrane system, which includes endoplasmic reticulum, Golgi apparatus, lysosomes, and the vacuole itself. Then we'll be also talking about mitochondria, plastids, ribosomes. These all are actually the cell organelles. And you have already learned about these concepts in your class 9th as well, right? You have been learning cell this chapter from class 8th, right? It was first introduced to you in class 8th, then again in class 9th, and again you're having this chapter in class 11th. So it becomes very important as per your NEET examinations as well. So we'll also talk about cytoskeleton, cilia, and flagella. What are these and what are their functions as well? We'll talk about what is centrosome and centrioles, about nucleus and microbodies. So if I talk about today's goal, basic introductory part, we'll have cell theory and the prokaryotic cells. So if I talk about the chapter importance, you have around 4 to 5 questions coming in the NEET exam, and 4 to 5 questions. That means you have a chance to score 16 to 20 marks, because each question, it carries how much? 4 marks. 4 marks in your NEET examination. And this is a very easy chapter, and you can easily earn those 16 to 20 marks if you're preparing very well. So let us begin. So talking about the great scientist, which is also mentioned in your NCRT textbook, which is G.N. Ramachandran. So he received his PhD from Cambridge University, 1949. Now, very interesting thing about him. Although his contributions are there in the field of biology, but actually he was an Indian physicist. Just imagine a person from physics working on the concepts of biology. So his work, it led to the creation of Ramachandran plot, which is a very famous one to talk about the intro about the structure of proteins. Right? It tells you what can be the conformation of various proteins. So he also proposed this triple helical structure of protein, that is collagen. You might be knowing about collagen, which is the most abundant protein that you have in the mammalian system or in the animals, you can say collagen. Now, talking about some of the basic terms that are associated with this chapter, I hope some of the students might be very clear about these terms. But even though if you are not, I'm here to help you out. So we'll be talking about what is a protoplast, protoplasm, cytoplasm, cytosol, and the nucleoplasm. Okay, so we'll be talking about all of these terms in today's class. So when I say a cell, right, let us say this is a plant cell over here. A cell will be having a nucleus. This is the nucleus, right? This is the mitochondria, which I have been showing here. You also have the endoplasmic reticulum. So what are these actually? These are the cell organelles that you have. You do have other cell organelles as well, but over here I have shown only these two. Okay, you have the nucleus. What is this outer membrane? This is the cell wall. Cell wall in the case of plant cells, right? You don't have cell wall in the case of animals. The inner boundary, the inner wall or protective layer, you can say, that is the plasma membrane. It is also called as the cell membrane or plasma lemma, PMCM or plasma lemma. Now, what I'm doing over here, if I just remove the outer covering, right? If I just remove the outer covering, that is the cell wall from here, what do I have, right? I'll be having the plasma membrane, definitely. Plasma membrane is still there. You have the nucleus as well. You have the endoplasmic reticulum and the mitochondrion. So what will I call this structure now? I'll be calling it as protoplast, right? So that means protoplast is a cell that is not having a cell wall. Easy enough, right? Let us move further. Now what I'm going to remove from this, let us find it out. So I am removing the cell membrane now. This is the CM or PM. I'm removing that also. What do I have? I will be having the protoplasm now. So I guess you guys are clear about these two terms now, protoplast and the protoplasm, right? In the protoplasm, you will not have this cell membrane also. Cell membrane is also absent in this case. Okay, so what is remaining? Actually, one more substance is there. You will be having some liquid content that is present inside the cell, right? Semi-fluid like content in which all these cell organelles are suspended. Remember that inside the cell, you have this liquid content in which all these cell organelles are suspended. Now what I'm going to do, let us see. What I have done, I have removed the nucleus now. Removing the nucleus, what do I have? I have the cytoplasm, right? So this is what cytoplasm is. You have the liquid content, right? You have the cell organelles as well. This is what cytoplasm is. Now let us see what is the next thing. What I'm doing, I'm removing the organelles as well now. Cell organelles are removed. What do I have? I only have the liquid content now. Now this is referred to as cytosol, right? I hope the terms are very clear. Let me just clarify this concept once more. So what I have done over here, I have taken the cell wall. First of all, I have taken a plant cell, right? What I have done, I have minus the cell wall from it, right? If I have minus the cell wall out of it, what do I have? Protoplast. Minus the cell membrane, I have the protoplasm. Minus the nucleus, I have the cytoplasm. Minus the organelles, I have the cytosol. Okay, so I'll be giving you some questions now based upon this. If you can do those questions, I guess your concept will be very clear, right? You might be knowing what is there inside the nucleus. Inside the nucleus, you have the genetic material that is called as DNA. Just remember that. Let us solve some questions then. So DNA replication is not possible in which of the following? Is it protoplasm, protoplast, cytoplasm, or the plant cell? Yes, you can answer this question. I'm waiting for a while so that you can think for a while and you can answer. Okay, so I'll be telling you the answer. So the correct answer will be option number C. That is the cytoplasm. Why, sir? Why cytoplasm is the correct answer? The question is saying DNA replication. What is DNA replication? Doubling of DNA, right? Whenever the cell divides, its DNA content also doubles. This is what DNA replication is. So DNA replication is not possible in the cytoplasm because in the cytoplasm, you don't have the nucleus. Nucleus is absent in the case of cytoplasm. While if you see the protoplasm, protoplast, and the plant cell, all of those will be having the nucleus. Okay, next question for you. Based upon the same concepts, endoplasmic reticulum is not a part of cytoplasm, cytosol, protoplast, or the protoplasm. So you get to solve these questions live on the applications, right? Once you join the ODA class, You'll be getting these kinds of questions during the live class so that you can have in-hand experience during the live class itself about your concepts as well. So the correct answer will be option number D. Cytosol is the correct answer. Because cell organelles, because endoplasmic reticulum, it is a cell organelle. It will be not present only in the case of cytosol. Next question for you. This is the final question. So cell membrane is absent in which of the following? Cell membrane is absent in which of the following? You have protoplasm, cytoplasm, both B and C. So the correct answer is option number D, both B and C. So protoplasm and cytoplasm, both of them, they will not be having the cell membrane. Cell membrane is absent in both of the options over here. So now talking about what a cell is, you might have been learning about cell. What a cell is? Cell is the basic structural and functional unit of life, right? This is the basic definition which each and every student knows. But I'm talking about a very general term. What a cell is actually, right? What you can say? A cell can be a small room, right? So I define cell as a compartment or the chamber. Any compartment or chamber which is separated from the outer environment, right? You have the outer environment in the cell, in your body also, right? In the cell you have the liquid content, the outside also you will be having the semi fluid content, right? So it is a compartment basically which is separated from the outer environment by a lipid layer, right? Why do we require a lipid layer? Why not something else? Because lipid layer is water insoluble, right? Wherever you have these cells, the outer environment is also a liquid one. If it is dissolved in the liquid, there is no like sense of having this lipid bilayer, right? If it is soluble in water. I hope you are clear about this term. So let us move forward. What else? Inside the cell, there are certain chemical reactions going on. Definitely, right? Inside your body cells, there are certain chemical reactions going on. We call them as metabolic reactions, right? Sum total of all the chemical reactions that are occurring in a living system. These are the metabolic reactions. You do have two different types of metabolic reaction: catabolic reaction, anabolic reaction. Chamka kuch yahad aaya purana, right? Catabolic and anabolic reactions going on, certainly. And what else? These reactions are carried inside this compartment. And what else? You'll be also having exchange from the outer environment. It's not like that cell will be living alone. It needs to be interact. It also wants to interact with the neighboring cells. It also wants to send some of the things which it has produced to the outer cells, or it also wants to receive something. So for that, there should be some gates, right? In your homes also, you do have gates, and the things can people can come in and go out from those gates, right? So similarly, in the case of cell also, you will have these gates, which are called as the membrane proteins. There are certain channels which allow and they also help in the transfer of these different types of transportation medium. You can say they act as a transportation medium. So this is what is happening in your body cells. So what is the requirement basically to make a cell? Number one is separation from the outer environment. Definitely you have this separation. In the case of plant cells, you have a plant cell wall, right? Inside that you have the plasma membrane as well. But in the case of animal cells, you only have the plasma membrane. So exchange from the outer environment, this is also very much important. The exchange should happen. The third thing is the genetic material. So a cell will be also having a genetic material which carries the genetic information, right? This is all about what you have inside the cell. So if you are talking about a prokaryotic cell, right? We do have two different types of cells now. One is a prokaryotic cell, that is a primitive kind of cell, right? And the newer version of the cell, that is the eukaryotes. What is the basic difference? So if you see a prokaryotic cell over here, it is also having an outer membrane, right? Outer membrane is also present in the case of bacterial cell, which is separating the inner contents with the outer one. You do have channels which can help in the transportation of various substances, right? There are chemical reactions going on, but what is the basic difference which you can see over here? The basic difference is compartmentalization. If you see a eukaryotic cell, there are several compartments also formed. These chemical reactions which were occurring just inside the cytoplasm, right? It is just occurring inside the cell. But if you see eukaryotic cell, there is proper compartmentalization as well, right? All these chemical reactions, they are occurring inside proper cell organelles. Now, these membrane bound cell organelles are absent in the case of a prokaryotic cell, right? In the case of prokaryotic cells, these will be lacking. Okay, so in the case of prokaryotic cell, what you see, the genetic material is present. The genetic material is basically naked, you can say. It is not present in a membrane bound organelle. It is just present inside the cytoplasm of the cell. And this space over here is referred to as the nucleoid. Where the genetic material in prokaryotes is found is referred to as nucleoid. But in the case of eukaryotic cell, what you find, the genetic material, it is present in a membrane bound system that is called as a nucleus. Okay, this is the basic difference that you have in the prokaryotic and the eukaryotic cell. So cell is the fundamental, structural, and the functional unit of life. The basic definition that you have, that means all the living organisms, they are made up of cells. So what is cytology basically? The study of cell structure is referred to as cytology. While cell biology, it will be dealing with the cell structure as well as its function and reproduction, right? So cell biology and cytology, basic terms that you have. And cytology is basically the study of cell structure, while cell biology, it also includes the study of cell structure, function, and its reproduction as well. So if you see, there is a proper level of organization in the case of human beings or multicellular organisms, which are made up of more than one cell, right? You do have unicellular organisms as well, but multicellular, which are made up of more than one cell, you do have a proper level of organization. So what actually happens? Initially, you have this one single cell, and this single cell, it divides to form a group of cells. Now, these are group of cells which are specialized to do certain function. These are referred to as tissues, group of similar kind of cells. These are the tissues. Then you have the organs formed from the tissues. These tissues, now different types of tissues, they'll come together and they'll work together to form different organs in your body. Once these organs are formed, what next? They'll be forming an organ system. For example, you have this digestive system. In a digestive system, you have multiple organs which are working together. Then you have in an organism multiple organ systems working together. And this is how a cell, it forms a complete organism. So what you can see, as you move from cell to organism, what you are able to see, that the complexity of the organism is increasing, right? Complexity increases as you move towards the cell towards the organism. So if you see, in the case of human beings, what happens after the fertilization? What is formed? Zygote is formed. That is also one single cell. And this single cell, it will be dividing to form the stem cells, which are the undifferentiated cells. These are basically the undifferentiated cells. These stem cells, they can form any kind of cells now, right? They have this potential to form any kind of cell in your body. And ultimately, what happens? You have different types of cells being formed. And these different cells, these are basically the specialized cells which can form various multiple cells like themselves. And these are what? These are called as tissues. Similar kind of cells which are specialized to do certain job. Now, cell can even exist in an independent unit. That is what we call as unicellular organism, right? Based upon the number of cells. Based upon the number of cells present in an organism, we can have two types of organism. One is unicellular and the other is multicellular. These organisms, they are made up of just one cell. And still, even though they are made up of just one cell, they are capable to do each and every function. Like they can have their own nutrition, they can excrete gaseous exchange. So all of those things which are needed for a living organism they can do. Even though you cannot see them with your naked eye, they are living organisms. So anything that is lesser than a complete structure of a cell, right? It cannot be a living organism. It cannot, if it cannot live on its own, if it is not having an independent existence, we cannot say that it is a cell. For example, you have mitochondria, right? Mitochondrion, it is having a double membrane structure. It also has its own genetic material, but then also it is not a complete organism, right? It is dependent on our body cells for its survival. It cannot live on its own. So this is the basic, you can say, concept of what a living organism is, what cannot be a living organism, right? If it is capable of independent existence, a complete structure of cell, it is a living organism. So talking about some important discoveries as well. So what do you have? Robert Hooke, 1665, he observed the first cell, right? Although it was a dead cell from the bark of oak tree plant. So he was the first one to observe the cell. And he also gave the term cellula, which means the Latin word for cell. Cellula means small, tiny rooms. And this was how the first cell was discovered. Then came Antony van Leeuwenhoek in 1674. So he was the first one to observe the living cells. So what he did, he made his own kind of microscope. With himself, he made this microscope and he was able to visualize the first living cells from the pond water. What he did, he took the pond water and he found these various living organisms like bacteria, protozoa from this pond water. So actually, he didn't know what to call them at that point of time. So he called them as animalcules. And this was how the living organisms, the first living organisms, were observed. Then came Robert Brown in the year 1831. So I hope you are remembering the dates as well. 1665, Robert Hooke. 1674, Leeuwenhoek. Then you have 1831, Robert Brown, who observed the nucleus for the first time. You might be knowing nucleus, a very important structure, right? Inside the nucleus, you have the genetic material in the case of eukaryotes. Prokaryotes, they don't have this nucleus. Prokaryotic organisms, they'll not be having this nucleus. Nucleus by 1831, Robert Brown. Then you have the discovery of electron microscope by Ernest Ruska and Max Knoll in the around like 1931. This was a good period, right? When we started seeing the inner details of the cell, right? This was possible only due to the discovery of electron microscope. And this was how we learnt about various other details about the cell as well. Then came the cell theory, right? You have these two important scientists, M.J. Sheldon and Schwann. They gave the cell theory, right? So Matthias Sheldon, he was a German botanist, 1838. So he was a botanist actually. So he was working upon these plant cells, and what he found that all the plant cells were composed of different types of cells. This was his conclusion. Then you had in the similar kind of time, 1839. This was a Theodor Schwann, British zoologist. So he was actually studying both plant cells as well as the animal cells. So what he found? He found that the cells, they have this outer thin covering that is called as plasma membrane. Plasma membrane is found in the case of all the cells. What else did he found? Let us find out. Very important concepts going on. So he also found that plant cells, they have cell wall in them. Apart from the cell membrane, they also have the cell wall, which is possessed by different types of cells. And what else he found? He proposed that animals and plants, they're composed of cells, right? So this was his conclusion, that all the animals and plants, they are made up of cells and product of cells. So this was the conclusion made by Theodor Schwann according to his research. So if there is a question like who proposed the cell theory, you will write both the scientists' name. Sheldon and Schwann both gave the cell theory. But if there is a question like who proposed that animal cells and plant cells are composed of cells and products, right? And their products, you will be writing Theodor Schwann. If there is an option like this, choose the option second one, right? Let us move forward. Now, there was a drawback also, certain drawbacks of the cell theory. What were the drawbacks? It was not able to explain how new cells arise. At that point of time, neither Schwann or neither skeleton, both of them were not able to explain how the new cells were forming. Then came Rudolf Virchow in the year 1855. So 1855, Rudolf Virchow, he modified the cell theory. What he told that all the cells, they arise from pre-existing cell. Let us say there is one cell over here, and this one cell, it will be giving birth or rise to a new cell. So this was explained by Rudolf Virchow. So this is the modified version of the cell theory, also mentioned in your NCRT textbook. All the living organisms composed of cells and product of cells. Second point, all cells, they arise from the pre-existing cells. So this is how the content is given in your NCRT textbook, right? All given in the form of paragraphs. So you have to decode it, right? So now talking about diversity of cells, you can have different types of cells, right? The longest cell that you have, in the case of animals, it's the nerve cell, which is also called as the neuron. It's having this elongated and long shape. Talking about the plant cells, in the case of plants, you have these fibers, right? Particularly called as these Ramis fibers. Neurons, this is how they basically look like, or the nerve cells, you can call them, both of them are same thing. The largest cell will be the ostrich egg if you are talking about an animal cell, while acetable for plants, right? This is how they look like. While the smallest cell in the animals will be mycoplasma. Basically, these mycoplasma, they're bacteria without a cell wall. They don't have this cell wall. Basically, a bacteria, either gram positive or negative, we'll be talking about these things, right? What are gram positive and gram negative bacteria as well. So if you talk about bacteria, they will be having a cell wall. But mycoplasma, they don't have a cell wall. In the case of plant cells, the smallest one will be duckweed plant. So this is how they look like. Now, talking about some overview, what is the size of cell mentioned in your NCRT textbooks? A typical eukaryotic cell, it can be around 10 to 20 micrometer. So you have these PPLO, which is also mycoplasma, right? Pleuropneumonia-like organisms. Okay. So the smallest cell, mycoplasma, you have 0.3 micrometer in length. The largest isolated single cell, that is the egg cell of ostrich. Size of bacteria, it can be around 3 to 5 micrometer, while the human red blood cell can be 7 micrometer in diameter. Shape of the cells, if you talk about red blood cells, they have this biconcave shape. Okay, so these red blood cells, they basically help in the transportation of oxygen. They're present where? In your blood vessels, and they help in the transportation of oxygen. White blood cells, these are having amoeboid shape. Basically, the amoeba, which is a single cell organism, they are capable of changing their body shape. Similarly, they can also change their body shape. So they're called as having amoeboid shape. Columnar epithelial cells, they'll be having this long and narrow shapes. Nerve cell, this is how they look like, right? Blanched, elongated and branched. Tracheids and mesophyll cells, they are found in the plant cells. They're present in the plants. So they'll be having these shapes over here. So we'll be talking about these two different types of cells in our next class. So thank you so much, students, for joining in. I'll see you in the next class. Till then, bye-bye.